Okay, guys, today we're going to talk about this new chapter entitled The History in Our Genes. Basically, here is what we're going to talk about today. We're going to talk about the concept of the genetic archive, and we're going to look into an experiment that was done by a guy that is very close to us, Dr. David Hillis at UT Austin. He proved the point, basically, of the genetic archive with an experiment made with viruses. We're talking about viruses every single day at this time, so this comes even more into consideration, okay, this experiment. We're going to talk about the concept of gene trees and the challenges associated with them. Then we're going to talk about neutral evolution, this concept, very simple, okay? And we're going to talk about the difference between natural selection and neutral evolution. Then we're going to end this discussion with the molecular clock. The concept of molecular clock really is a concept which also helped understand the age of the HIV virus. We're going to talk about viruses quite a bit in this lecture, and again, now more than ever, you will be interested, I hope, in knowing more about viruses and how they behave. I don't know, many of you recall, we have been talking about viruses quite a bit to give examples. We have talked about the fact that they mutate very quickly, and now you can understand how important is this concept. Now that we are seeing with our eyes, we are experiencing how powerful is this strategy of viruses that they have to mutate so quickly, so much so that they can even jump species. We are dealing with a virus that most likely has been jumped from probably, they say, a bat reservoir, so wild animals, that's mutated enough to where now it can infect humans, okay, human cells. And this is true for many viruses that we have had in the human history, if you will, including the HIV virus, which we're going to talk about at the very end. I'll tell you everything that I know about the HIV virus, its history, its origin. Again, it was a virus that infected chimps, monkeys, and a jumped species, just like the coronavirus, okay? So we already know, we have this concept of viruses mutating quite a bit, quite fast. All right, so let's start with the beginning, okay, with the genetic archive concept, and we move on to these concepts, okay? The experiment of David Hillis, you have to be able to understand and describe, recognize. It's definitely going to be 1 point asked probably in a test, okay? So first of all, we have already encountered the concept of homology. If you remember, we defined very well this term, homology versus analogy. Homology refers to any characteristic and typically is a physical characteristic, for instance, a bone set, that allows us to understand if organisms have common ancestry, basically. Whenever we see structures that are very similar or pretty much the same, just change the shape a little bit, such as the bones in a forelimb of a human and a wing of a bat and a wing of a bird as well. Basically, as you see here, color-coded, these are the same sets of bones that we recognize. So we know that these organisms must have a close ancestry, a close ancestor. And so we have been acquainted so far mostly with the concept of morphological comparison, anatomical comparison of structures. But as much as that is true, we also have a so-called genetic archive, which compares homologies in DNA. How similar, these are sequences, don't get scared, this is very compressed. If you zoom in, These are sequences either refer to comparing sequences in the DNA or in proteins as well of different organisms. Okay, and so you know that DNA is composed of 4 different elements. You know, we can compare them to 4 different letters, and the sequence of these letters, specific sequence of these letters, identify specific genes. And we can nowadays we can very easily sequence the entire genome, which is the entire set of genes that compose the genetic material of an organism. We can there are programs that align these sequences for us and highlight the differences as shown here. Okay, and so we can understand how similar is the genomic material of different organisms. And as much as we do that for structures, we do that for DNA, for molecules, especially DNA, but many times also proteins. It's the same thing. You're comparing structures, you're comparing sequences, okay, to understand what is the degree of homology, okay. And this approach can lead us to construct evolutionary trees, okay. Remember that historically evolutionary trees, phylogenetic trees, have been constructed using morphological comparisons of anatomy of structures. And we have already learned at the very beginning. I hope you guys remember that. If you don't, you have to revise it. Remember that the last test is going to be comprehensive. We can also compare organisms at molecular level and to understand how closely related they are. Okay, this is referred as the genetic archive. Now, then regarding the genetic archive, this is an experiment, a brilliant experiment that was performed by Dr. David Hillis at UT Austin, so pretty close to us. He actually, in order to prove the point of the genetic archive, to prove the point that you can look at mutations, which are differences, right, in the same genes of different organisms, because, you know, we share pretty much all genes with, especially with the other primates, but also all the other vertebrates. And there are even genes that we pretty much share with bacteria. So very, very fundamental genes, and therefore we can compare these gene sequences to understand how far apart two groups are. And of course, as you can imagine, the more mutations we see, the more differences we see in gene sequences between two species or two groups, the more distant they are in terms of time, on a timeline, because over time there have been mutations that have accumulated during evolution. So that's the concept. Now, this is a concept that is pretty easy to grasp, I guess, but David Hillis did an experiment to prove this point. Now, the one thing that, of course, is impossible to do is to use organisms, especially complex organisms or organisms that take quite a bit of time to reproduce, to prove a point like the genetic archive, because of course it would take too much time to get enough generations, enough change over time to prove a point like this. But smartly, David Hillis utilized actually viruses, and there are some viruses that are specific to infect bacteria. And these viruses that infect bacteria look just like this drawing, look like spiders, where in the head they conserve their DNA, which is the genetic material, the message that says all how these, I cannot call it organism because viruses are not living organisms, but how this entity needs to appear, needs to be reconstructed. Okay, so viruses basically are non-living. There is no cell here, no cell structure we can recognize. A virus is an envelope. This envelope, in this case, looks like a spider, which contains a genetic material, a stretch of genetic material which has the information on how to make new copies. Now, in order for this entity to replicate, to make copies of itself, it needs to get into a cell because it has nothing with it to otherwise make copies of itself. Okay, so that's a virus. Now, let's look at what happens when a virus that infects, for instance, E. coli, bacterium E. coli, this is called the bacteriophage specifically, and the T7 is just the strain that was used by David Hillis in his experiment. So don't worry too much about remembering the name of the specific virus, but at least to recognize it if you see it written in a question. So let's see what happens when a virus infects a bacteriophage, specifically infects a bacterial cell like E. coli. First of all, the virus does not really penetrate into the cell. All it does is attaches to the cell and injects the only important thing that it's necessary to make copies of itself, which is that message, that DNA that is encapsulated in the head of this virus. Once this DNA is inside the cell, it has all the information to kind of like hijack the cellular machinery and make copies of itself. Okay, that's it. These are many copies. Now, once all these copies are made, this stretch of DNA contains genes. Genes make proteins, and so this DNA, this or replicated DNA, instructs the cell to make all the proteins that are necessary to reconstitute all viral particles, which are represented here. Once lots of particles have been reproduced and copied, now the cell is full of this viral particle, and what happens is that determines a cell explosion that is known as lysis. Pretty much the cell dies. Unfortunately, this is also what happens, of course, when many times human cells are infected with viruses. Okay. Okay, so and this particular very aggressive type of cycle of viruses is known as lytic cycle. In reality, there are also viruses that instead of going directly through this very aggressive cycle, they go through what's called the lysogenic cycle, which is basically once the DNA is inside the cell, this DNA, what it does, it's kind of introduces itself inside the host, the genome of the host, the DNA of the host, and it can stay kind of quiescent. It's there, but does nothing. This can happen, for instance, a lot. It happens a lot with the HIV virus, and only at a certain point, maybe because of external stimuli, it starts really replicating, okay, and then determines the lysis of the cells and the death of the cells. Okay, so, but this is what specifically happens with bacteria. So I'm telling you all this because, of course, it's important to know this, to understand the experiment that David Hillis performed in order to prove the point of the genetic archive. So the other thing I want to show you is, in reality, what does it mean when these cells lyse and die? Here, I'm showing you a Petri dish. I don't know how many of you have ever seen a Petri dish. These are dishes that are used in the lab to culture many things, including bacteria. In this case, there is a jelly material here. It's called agar, on which a carpet of bacteria, you cannot see them really because it's like the whole plate, the whole Petri dish is invaded by bacteria, but it looks very opaque, okay? Now, when you infect this plate with viruses and the viruses determine the lysis, the disruption of many cells, you end up with a plate with a petri dish or plate that looks like this, where you see all these areas where there are no more bacteria. Why? Because they've been destroyed, they've been lysed by the activity of the virus, the replicative activity of the virus. So you know that this plate has been very well infected and then the virus has multiplied. And so you know that there, and every time actually there is a multiplication and infection and replication of a virus in bacterial cells, there are up to thousands and thousands of copies of the virus are produced. Okay, this is just a schematic representation. It shows you a few, but it's much more than that in one cell. And so this gives the opportunity to the virus to mutate very fast. Not only the viruses have the ability to mutate very fast, but the fact they can produce many thousands of copies of themselves in one infection cycle, it gives them even more opportunity to mutate. Okay, so this is the concept that you have to understand in order to understand the experiment that David Ellis performed. Okay, so this experiment was carried out in 1992 at UT Austin. David Ellis demonstrated how to read the history that is coded in genes. And he used the bacteriophage T7, which looks like a spider and infects E. coli cell specifically. And as I showed you, the infection can be tracked by looking at the formation of these plaques with these areas that kind of like where the bacteria disappear on a petri dish. So what did David Ellis exactly do? He did a series of infections. That's all he did. And then he analyzed the DNA of the viruses. And this is how schematically we can represent the infection cycles that he produced. First, he started with the first plate of infected E. coli, okay, where he had those plaques that I showed you. And this was his kind of like original population, okay, his original lineage, or we can say the ancestor plate, okay. This is represented as a phylogenetic tree, okay. This is a whole clade. And what he did, from the very first plate, which is represented again here, he then split this viral population basically in two. One he kept there, and the other he split in other two plates. He infected two different plates to create to resemble what happens in speciation, in the formation of two different species or groups, we can say, in a phylogenetic history. Then he did that again. With this plate, he infected two other plates, and then two other plates. Okay, and then at some point he stopped. This is a schematic representation of how many times he did the split and how many plates he infected. All these plates that you see at the tip of the phylogenetic tree represent like the final lineages. You can compare them to final lineages of phylogenetic tree. And then he went ahead and he actually tracked down the mutation. And here is shown how these two plates, which came from a common ancestor here that had been split in two, they had the same, actually the same mutation. So he was able to demonstrate that, yes, when you are looking at mutations of lineages and you find homologies in these mutations, you can track down these mutations to a common ancestry. That's what he actually proved in this experiment. And his experiment didn't last very long because a viral infection is very quick. If you wanted to reproduce this concept, even with bacteria, it will take probably years, and you could never even do it with with multicellular organisms. So by using viruses, which, by the way, as I told you many times, are not even considered living organisms, but the concept of mutation and the genetic material they carry is the same as ours, as all the living organisms. So you can do this kind of experiment to prove a point, okay, on how mutations can be tracked down to understand a common ancestry and to build up a phylogenetic tree. All right, so this is the point that he pretty much proved, okay? All right, so I already explained to you everything. Pretty much here is all like blah, 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 what I just said, okay? How where all the lineages were split over and over, and what were the conclusions, okay? Now, so the conclusion of all this, okay, is that relationships of species and other clade groups can be inferred from their DNA as well as their morphology. Okay, so we nowadays we know that we can rely on molecular analysis, differences and mutations in order to understand phylogeny, not just morphology as we have done historically. But we have talked about basically this concept already at the very beginning. We are recalling it and we are just talking about it in a little bit more detail. All right? Okay, so now I want to open your eyes to one thing. We talk about differences and comparisons in DNA, okay? Imagine that DNA is only made of 4 different things, 4 different nucleotides. We identify them as bases, 4 different bases, which is the variable part of a nucleotide, of course. And so we can talk about 4 different bases. I know this is hard for you to understand, but just compare DNA as to a code made of 4 different letters. You can call them A, G, T, C, okay? That's fine. So only 4 different letters, that in all kind of combinations. And remember, genes are thousands of bases long, and therefore, obviously, you can have so many different combinations of these 4 letters, and they can be also repeated in some stretches. So consider, for instance, that the human genome contains about 3.5 billion of these bases, okay, of these letters, all in different combinations. Now, there are some challenges. The use of molecular trees is great. It's a great tool, but it's not all golden. These are the challenges I want to point out. First of all, and this is, I think, something we have talked about at the very beginning as well, DNA decays rapidly after death. And so basically this makes nearly impossible, to compare DNA from extinct species, from remains of what we find extinct species, while it is kind of relatively straightforward to compare, for instance, skeletal or whatever is the remain, if it's a shell, the shape of a shell, or bone remains from creatures and species that have lived a long time ago that are extinct. Now, the other problems are parallel substitutions and back mutations. What are these two? Substitutions, you can also call them mutations. We could say parallel mutations and back mutations. We call them substitutions right now. That's another term. Parallel substitution refers to the fact that some groups will get the same mutation independently. Being the fact that you only have 4 letters that compare DNA, okay, that form DNA, This means that two lineages that you're looking at may have acquired by pure chance the same mutation, but this does not mean that the two are necessarily coming from common ancestry. It could be that it was just a case, it was just something that happened just randomly by chance. So that's one caveat here. The other caveat is known as back mutations. You cannot exclude that during evolution, during millions of years of evolution, in lineages, you have had a mutation which changed the one letter into the other, maybe A in C, but you cannot exclude that the C reversed to A again by mutation. So back mutations. So, and now you are comparing, for instance, two lineages that present both at A, that position, and then you get the idea that these two lineages have common ancestry. They're very close together because they show the same sequence, the same letter at that position, but in reality, that has been the result of back mutations. So you have to keep in mind all these challenges, all these problems. And scientists that, of course, are in this field do keep in mind these things, okay? And the best thing to do here is always to compare more than one gene, many genes together in order to understand how closely related are groups. And what is true is that the molecular trees that have been the gene trees or molecular trees, or whatever you want to call them, many times they recapitulate 100% the morphological trees that have been built in the past. Okay. All right, so keep in mind all these challenges. Now, the one particular challenge I want to talk about is the evolution of humans, chimps, and gorillas. This was for some time very controversial. We didn't know who was the group that was more closely related to humans, chimps or gorillas. They all have, these two groups have like a lot of homology in common. The genome has a lot of homology, same degree of homology overall to human. And so the question mark for a long time was how chimps or gorillas more closely related to human. And in order to understand and to answer this question, let's look at a scenario, okay, that could be misleading, okay? Just to understand how important it is to compare many genes, not just one or two or three genes. Let's look at this scenario. This is also an example given in your book. These are different scenarios of different alleles. You know what an allele is at this point, a gene variant. Let's look at different genes, gene A, gene B, and gene C. Gene A, gene B, and gene C, and build up a molecular tree, a gene tree, based on just the single genes, A, B, and C. Now, gene A, we imagine has only one allele. Gene B, imagine that has two alleles represented as green and blue. And gene C also has two alleles represented as green and blue. If we look at gene A, it seems like our question cannot really be answered because it's found unchanged. The dot represents the allele not changing or not being lost in the three groups that we are comparing. These are really three species. So gene A does not help us understand who is more closely related to whom. And I know that here represents like chimps more closely related to humans because actually we know that nowadays, but we know that from other studies, not from gene A. Let's look at gene B. If we looked at gene B only, it seems like the blue allele, let's start from the common ancestor here, it's passed also to human, chimps, and gorillas, as well as the green one. So again, gene B that has 2 alleles has also been passed equally, so we cannot draw a conclusion from gene B as well. But let's look at gene C. Gene C has again 2 alleles. The blue, let's track the blue. The blue is found in humans, unchanged with the dot, is found in gorilla, unchanged, but oops, it stops in chimps. And let's look at the green allele. The green allele, it stops in humans, it stops in gorillas, and it keeps going in chimps. So by looking at gene C and the 2 alleles, the behavioral 2 alleles, it seems like humans and gorillas have this much more homology than humans and chimps. So from gene C, you could draw the conclusion that actually humans and gorillas are more closely related. So actually this phylogenetic tree should have the gorillas over here, okay? In reality, it doesn't, because we know nowadays that this is the correct tree where humans and chimps are more closely related. And how do we know that? In order to understand this, we had to resort to the analysis of some sequences in the human genome as well as chimps and gorilla genome that are not really genes. These sequences are very mysterious. This is very fascinating, guys. And this is something that my students struggle to remember for some reason, but I'm telling you, this is important. It's going to be in the test. So, it's sometimes, in order to understand how to solve these questions, to answer these questions, such as which is now the group more closely related to human, you don't look at genes, because genes don't help you. Remember this example we gave before? There's too much similarity. You cannot look at genes. But I don't know if I ever told you, I can't remember, to be honest, right now, that our DNA, if you look at our genome is not made of mostly of genes. Actually, our DNA is made mostly of what is called junk. Basically, our DNA is full of meaningless sequences. I'm telling you about billions of those letters that don't represent genes, don't represent anything. They're called junk. And while many of these sequences nowadays we have discovered they're not really junk, they help regulate the genome and the production of proteins, but still there are many sequences that are pretty much meaningless. And I wish we were in class, because I would explain this concept a little bit more in detail, but just take it as it is. It's just a fact, really. Okay. Now, among this massive junk, remember what I told you at the beginning about viruses? Some viruses are sneaky. They don't just get into a cell of a host and start replicating. They're able to sneak and to insert into the genome, into one chromosome of the host, of a cell of a host. This is what HIV virus does, actually. And so that virus becomes part of a chromosome, part of the genome of the host. And this is how many viruses behave. You know, viruses are very ancient. There is a theory out there that says viruses are even more ancient than cells. So as you can imagine, viruses have been around for a long time, probably even before life appeared on Earth. And is it possible that a species, that an entire species gets a virus inside the genome, and this virus gets passed on? Remember, virus as just a sequence of DNA could be passed on to the next generations. Yes, it's possible. When we look at our genome, there are many viruses, many ancient viruses that are in our genome there, they're kind of like they've lost their purpose of being viruses. They've become part of the massive junk of our genome. And some of these sequences are called ALU elements, okay? That's it, just a name. These ALU elements, which were studied by Mark Batzler, this is just a scientist, again, are thought to be basically ancient viruses that don't have the function of the virus anymore, they're just there. And there are many of these sequences, many of these ALU elements that are present in human as well in chimps and gorillas. So can we look at these sequences? If a common ancestor before the split between humans and gorillas, or humans and chimps, had this virus, it will be passed on. So it's like almost like a gene. It's not a gene because it doesn't produce a protein, but it's a sequence that is in the genome that it keeps being passed on. So could we use these elements, these ancient viruses in our genome and the genome of primates, to kind of compare the mutations that happen there to understand which group is more related to which? Yes, of course we can. And that is the work that Mark Batzler did. He actually studied 33 ALU elements. You can call them ALU elements if you want, and found in gorilla, chimps, and humans. This strongly suggests that they were inherited by a common ancestor. Okay, so you have 33 ALU insertions. They're called insertions because they're insertions of viruses, right? Or you can call them elements, that's fine too. And of these 33, he discovered that actually 7 ALU insertions are shared only by human and chimps, okay? And 7 only by humans, 6 only by chimps. And so basically based on the comparison of the ALU insertions and their mutations, Batzler was able, and his group was able to strengthen the hypothesis that actually was already out there, that human and chimps are more closely related to each other than either is to gorillas. Okay, and so there was a diversification of groups. One group from a common ancestor, one group became gorillas. The other group split again in the group of humans. And humans, you'll see we're not just talking about Homo sapiens, but the whole human Homo. And we'll talk about the whole Homo group later on in the course, and chimps as well. All right, so this, again, the concept of ALU elements, what they are, and they helped understanding which group is more closely related to you and between chimps and gorilla. This is something you must know. All right, it's just, again, a concept, and this is our most closely, this is our uncle, okay? This is uncle bonobo. This is dwarf chimps. These are the chimps, the group of chimps that are thought to be the ones that are most closely related. There was a split here to humans, okay? All right, so as I told you, molecules and morphology, molecular and morphological trees may overlap each other very perfectly. And this is in most of the cases. Sometimes molecular trees make us revisit morphological trees. And this was the case of the domains. Remember what I told you at the very beginning about the 3 domains? That initially we were actually considering archaea and bacteria splitting from a common ancestor, and then when we had a molecular tool, we had to revisit that very first, very general, broad phylogenetic tree, and actually we discovered that the molecular level archaea are much more closely related to eukarya, and so we had to put them splitting from the same branch. So that was the case in which we had to modify a phylogenetic tree. But for instance, I want to give you an example of the phylogenetic tree of tetrapod origin. Remember, tetrapods come from lobefin fishes. We studied this. Coelacanths and lungfish, which are the two living groups of lobefin fishes. And when scientists compared DNA and molecular level of all these group, tetrapods, the two living lobefin fish, as well the rayfin fish and cartilaginous fish, they came up with this kind of phylogenetic tree that is the same phylogenetic tree, exact same phylogenetic tree that had been constructed upon morphology. So as you can tell, many times molecular trees reinforce and confirm morphological trees. Okay, so we have done a good job. Okay, so fossil remain made a prediction and DNA supported it. Okay, we know for sure that lungfish were the closest relatives of tetrapods. Remember when gas bladder became a lung, and then this gave rise to tetrapods, along with the evolution of a lobefin, All right, so the molecular tree has also been very useful to understand human evolution. We know for a fact that the oldest fossils that are closely related to Homo sapiens have been discovered in Africa, and they're about 200,000 years old. This is a fact. This is a fossil evidence. The next oldest fossil relating to Homo sapiens are from Israel, actually, and they are about 100,000 years old. And younger fossils have been then discovered in Europe, Asia, and Australia. We will talk about human evolution, Homo sapiens, where we come from, and all the hominidae, the major hominidae groups, actually in the last lecture of this course. Now, we historically have been comparing fossils. Okay, we have fossil evidence, and we have constructed our phylogenetic trees and hominidae history on that. We know that because, especially because the oldest hominidae, I told you there are 200,000 years, Homo sapiens, 200,000 years old, they were discovered in Africa. So the Carl Stringer, a known paleontologist, he argued that humans, Homo sapiens, originated in Africa, and then it starts spreading out to other continents. We will talk about this. This is not just a theory, this is a fact. So whether you like it or not, we all come from Africa, okay? This is where Homo sapiens appeared. This is a fact, it's been pretty much proven, not only from paleontology, from molecular evidence, from morphological evidence of fossils, but also has been confirmed by molecular analysis, because there is one type of DNA in our genes, is the mitochondrial DNA. There is DNA in our mitochondrial, the powerhouse of cells. There is a type of DNA that is much more resistant than the DNA that is in our chromosomes. And this DNA preserves much better than the chromosomal DNA, and is usable to sequence even with having remains that are so ancient. And so by looking at mitochondrial DNA of fossils, and also by comparing the mitochondrial DNA of living Africans and all the Europeans, all the other groups of Homo sapiens, we can say, that have conquered other places over time, evidently. What they saw is that when they compared actually the mitochondrial DNA of living Africans, they saw that Africans carry the biggest diversity of mitochondrial DNA, and then the various groups that have come later in Europe, in Asia, they have a much less diversity, as well as they're more similar to one another. And so this is evidence that in fact Homo sapiens was born in Africa. Okay, and so mitochondrial DNA again has been useful to kind of understand what the history of Homo sapiens is, and that it appeared in Africa and started migrating out. But also, as I told you, the fact that mitochondrial DNA is much more resistant to degradation than chromosomal DNA. We have also been able many times to actually collect samples of this special DNA from fossils and compare it directly. Okay, so that also has increased the support of this idea. Anyways, as I told you, we will talk about this much more in detail in the very last lecture of our course, but you can start remembering this for me. That's great. Okay, and this is kind of like a timed history of migration, okay, where you have first appearance in Africa, and then about 200,000 years ago, Homo sapiens appeared, and it took some time for him to start migrating, okay, and this is the migration pattern, okay. About 100,000 years ago was already in an area like in the Mediterranean area, such as Israel, and then slowly migrated everywhere else, okay. And when we talk about hominidae evolution and the history of migration of Homo sapiens as well, we'll talk about why we do recognize differences in phenotype of all the groups of humans that nowadays have, of course, migrated everywhere in the world. All right, so now the last concept really of this lecture is the concept of neutral evolution and a molecular clock. What is neutral evolution? Okay, we talked about natural selection. We talked about natural selection, that is a process of change over time of species along the course of generations, of course, because of mutations that determine something different in the organism. Remember, we talk about changes. Ultimately, we're talking about mutations. We're talking about changes at the genetic level. Genes produce proteins, and therefore, we're talking about different proteins here. Now, when these changes create or cause change, some sort of change, then the environment can select in a positive or a negative way for these mutations. But many times, mutations that happen in genes, even though they change the sequence of a protein or a sequence of the gene, perhaps they don't really affect the functionality of that protein. And these are known as neutral mutations. Now, as you can imagine, these neutral mutations are not going to be selected in a positive or a negative way by the environment, because they're just neutral. They don't really change the functionality of that protein. They don't really change that organism. They're just neutral. So neutral mutations are just going to drift around randomly. They're not just going to be selected for, because they don't affect the fitness of an organism. So based on this concept, okay, we know that when we look at proteins in organisms, different species, we have many of these neutral mutations here. They're represented as, Remember, DNA and proteins are made of a single, we call them monomers, unit, okay, of things. In DNA, they're nucleotides. In proteins, they're amino acids. It doesn't matter. But when you look at the protein, a protein, for instance, in a horse, in a human, and a gorilla, for instance, we're talking about hemoglobin here, you'll see that indeed there are some differences. The differences are given by the different colors. Look down here, there is a difference, okay? But if this difference in the hemoglobin, which is the protein that carries oxygen, they don't affect the ability of that protein to carry oxygen, they're neutral, okay? And therefore, they may be conserved, and maybe once they appear, they're not selected for, and they easily sneak into a species and they're carried on. So we could look at these neutral mutations, perhaps, in order to understand how closely related are groups. Okay, so this is the concept that we will talk about in a minute, about the molecular clock. For now, remember, there is natural selection we talked about extensively at the beginning. Neutral evolution refers to the appearance of these mutations, which are neutral. And because they're neutral, they don't affect the fitness and they don't change the functionality of proteins, they're going to be maintained in a population. They can be, we say that they're fixed, okay? And this happens because of genetic drift, not because of natural selection. But the concept of neutral evolution allows scientists to estimate the age of common ancestry of groups, or in other words, when did these two groups diverge from one another? And so this is the concept. Now we can use this knowledge to introduce the concept of molecular clock. It has been observed by scientists that neutral mutations accumulate at a roughly clock-like rate. So basically, we can look at, again, neutral mutations to understand how, when is the time of split of two lineages, of two groups, of two species. And there are many proteins, as I told you, that are common in pretty much even from bacteria to human. There are such basic genes, such basic proteins. So we can look at proteins, all kind of proteins that are common in organisms, to understand how, to look at neutral mutations to understand how far apart they are, basically. And this is a graph, for instance, of how this method, this molecular clock method, based on number of neutral mutations, are called substitutions. For instance, here is a cytochrome C that is used. The cytochrome C is a very essential enzyme molecule in metabolism, and is present in all these groups shown here. When we look at the number of neutral mutations or substitutions, we can actually set a graph like this, where time of split, or some time since the last common ancestor in million of years, is the representative of the x-axis, and a number of substitutions of that particular protein we're looking at, in this case is cytochrome C, but it could be hemoglobin, for instance, represented on the y-axis. And by looking at these numbers of substitutions, we can deduct, there's kind of like a correlation, okay? For instance, this is 75 substitutions, 75 neutral mutations correspond to about 80 million of years of split. So this would be, for instance, the time of split, or how far apart are humans and rodents, or dog and cow. They're kind of like similar, okay? So we can, again, build up our graph, and then from counting the number of neutral mutations or substitutions in particular molecules, we can kind of infer the time of split of two groups. And so this is the concept of molecular clock. And of course, we're not going to look just at one protein, but many proteins, and maybe average out our data. Okay, of course, these are very complex methodologies. They use supercomputers to do this analysis. We are far from describing this in detail. This is just a concept, okay, that I'm asking you to recognize. All right, now, as I told you, molecular clock, reading the molecular clock is not easy. There are also problems associated with the molecular clock, because we assume here that there is a kind of like a steady state of change in of accumulation of neutral mutations over time. But this is not necessarily true. There could be periods of time where rate of mutation is faster or slower. So, of course, there's a lot of error in these things. But again, they're still very powerful tools to kind of like infer, you know, in a certain range of time, the timing of a split of groups. All right, now, based on molecular clock, I want to tell you how we have been able to understand the age of HIV virus using the molecular clock. Again, guys, this is a virus, but it carries, actually, to be precise, HIV virus as well as coronavirus doesn't carry DNA as genetic material. It carries RNA, which is a little cousin of DNA. We're not going to go into the details of that, okay? But it's genetic material, okay? I just don't want to tell you there is DNA in this virus when it's not true. It's RNA, okay? But it's genetic material. Doesn't matter. It's a very similar molecule to DNA, still genetic material. This is also the case of the coronavirus. It carries RNA. In fact, I don't know if you have heard that some people have been treated for coronavirus using the antiviral agents for HIV. And the reason why they may work to a certain extent is because these are both RNA viruses. Anyways, this is just a curiosity. I think it's important that we also talk about real times. Okay, we're not just talking about something abstract here. You can understand how real our virus is now, especially at this time. Anyways, HIV virus, I hope that you guys, just like me, you're very scared of HIV virus. This is a virus you should be scared much more than the coronavirus. Because this virus, although we do have some drugs nowadays to kind of slow down its replication, is not killing people right now because the cocktail of medications that has been developed keeps it in check. But in fact, what you can read now is that many people are starting to be refractory to these medications because the HIV virus is mutating. It doesn't mutate as such as very fast rate like the coronavirus, thankfully, but it's mutating. And so maybe at some point, who knows if these drugs that have been developed are still going to be effective. Remember, there is no vaccine for HIV virus. So anyhow, what I wanted to introduce to you is the concept of the fact that we have been able to use the molecular clock concept that I just explained to you to understand the age of the HIV-1 virus, which is the most common strain. There is also another strain, HIV-2, and HIV-1 is the one that comes from chimps. HIV-2 is the one that comes from monkeys. And oops, I have already revealed to you where HIV comes from. Yes, HIV used to be SIV. HIV stands for Human Immunodeficiency Virus, number one. And it used to be SIV. S stands for simian. Simian, Latin, means monkey. So HIV virus is a virus that has jumped from chimps and monkeys to humans. Just like the coronavirus has jumped from, most likely, bats to humans. By mutating, it's been able to change these proteins that you see on, these are proteins, they're spikes that you see on the surface of the virus, that now are able to bind and be recognized not anymore by monkeys or chimps, but by human cells, and therefore they're able to penetrate a human cell. So molecular clock has been used to look at mutations of 159 HIV-1 viruses and understand when HIV virus we think that appeared, or that jump was made between chimps or monkeys and human. Because, you know, the HIV virus, well, we started seeing people dying mostly in the 80s. But does it mean that it appeared in the 80s, or it may have appeared a long time before? Now, remember, HIV virus is not like coronavirus. HIV virus needs direct contact to be transmitted. So obviously it's much more difficult to transmit. And also we're talking about period of times where people weren't traveling as much as they travel nowadays. So of course the spread was slower, but yet it happened. So the fact that we saw most people getting infected in the 80s, here's all samples, blood samples, people dying for HIV collected, and HIV virus analyzed, and here shows it shows a phylogenetic tree where here all substrains of HIV-1 with the specific mutations. Okay, that's what this diagram is showing you, and here is the years from 1980 to 1994. These are all the mutating strains that were identified. And by looking at those mutations, scientists were able to also understand what is the rate of mutation in this virus. And based on that, using very sophisticated analysis and computational analysis, they were able to estimate, remember this is all what was analyzed, that the virus was in fact born, and the mutation that allowed the passage of species happened around 1931. Okay, so you can understand now how, and this is a very powerful knowledge. And actually, I shall tell you that of course, all these samples were collected from people in the US, in Europe, but one sample, when they did the study, one sample was not included, and it's a sample that from a man that died for a very strange illness. Remember, HIV gives you very it deprives you of your immune system, so you end up dying for very, if I can say, stupid diseases where normally your body is able to combat this. Anyways, a sample coming from a man from the Republic of Congo, had been stored from 1959, was kept at the hospital, and then when it was later analyzed, when they understood about this epidemic of HIV, they determined that this guy had in fact HIV. This is considered the first person to basically where the virus jumped. So when they reanalyzed and did all the comparison again, including the sample that had been excluded initially, they now said that the first infection, in fact, had happened in 1959. Anyways, much earlier than when you saw the epidemic. Just wanted to spend a minute on HIV. We haven't learned much lessons apparently, because the reason why HIV virus, there was an SIV virus, was in chimps and monkeys, was able to jump species is for the very same reason that coronavirus jumped. It's because people were hunting and eating and cutting up wild animals. And we have learned a lesson, because the bushmeat tradition, which is the tradition of going out in the bushes and capturing anything from, I don't know, lions and chimps and monkeys, cutting them up and eating them, that was a tradition in Africa. And I don't know how much is still going on. And that's the factor thanks to which HIV virus jumped species. And not because the virus was ingested, but remember, when people capture an animal, they cut it up. And so by cutting it up, a mixture of blood between the blood of these animals and the blood of this person happened. And the virus had the right mutation. It just takes really one person to start an epidemic or even a pandemic. And so that's what happened, okay? And again, in just a few months ago, this happened again in China, in Wuhan. And now this is the reason why I am talking to a computer right now, because there is a pandemic because of the use of wild animal that are cut up and eaten by people. I know that the wild market of Wuhan is being closed and it will never be reopened again, I heard, and I'm very happy about that. This does not exclude that people may still go out and capture wild meat. But again, this is a lesson that we should have learned a long time ago, and we haven't, as humanity. Okay, so it's the same story that repeats itself. Okay. All right, so to conclude, molecular clocks are a useful tool that can definitely make us understand how evolution has taken place, and even to track down the age of viruses and when lineages have split. Okay, so I have to tell you there are some hidden slides in this lecture, which I'm not going to talk about. This was more of curiosity stuff. I'm sorry. I'm sorry. Here from slide 40, disregard these slides. I have them there just because when I have time, I like to talk about HIV virus, how it works, and things like that. But I'm not going to do that in this recorded lecture, okay? So these are the concepts that we talked about, and in this lecture, okay, from the Genetic Archive, the experiment of David Hillis, gene trees and their challenges. We talked about neutral evolution and the molecular clock, which is actually looking at the neutral evolution, and we talked about the history of HIV. All right, so I'm done, and I will see you next lecture. Thank you.